My name is Marissa Brinkmann, and for my biology role model project, I researched Jennifer Doudna. She is an American biochemist and the co-founder of CRISPR-Cas9. At the age of 7, Jennifer and her family moved to Hilo, Hawaii, and she grew up as a Hilo, which is the Hawaiian slang for outsider. Because she was considered an outsider, she felt alone and isolated at school, which drove her to take risks and prove doubters wrong. As a child, she read books to learn about the world around her and explored the biological diversity in Hawaii. She was curious about the world around her and captivated by the collaboration of scientists to solve biology's puzzles. She was inspired deeply by Rosalind Franklin, whose X-ray crystallography image captured the structure of DNA. It was the first time that she realized women could be great scientists. Her curiosity led her to pursue a career in science. She received her undergraduate degree in chemistry from Pomona College in California. While she was there, she completed research with Sharon Panowsko, where they studied the chemical signaling that enables bacteria to self-organize when they are starved of nutrients. It was the first time her name appeared on a research paper. She continued her education at Harvard University, where she received a PhD in biochemistry. During her time there, she completed research with Jack Szostak, involving self-replicating RNA and the potential role it had in the origin of life. She completed a postdoctorate at the University of Colorado Boulder, where she continued her research on RNA with Tom Scheck. She used X-ray crystallography image to image RNA enzymes in hope of maintaining a molecular portrait. After her postdoc, she accepted a position as an assistant professor at Yale University, where she continued her research on the basic structure and functions of RNA. She then accepted a job as a professor and faculty scientist at the University of California, Berkeley, where she continued RNA research that led to the discovery of CRISPR. Her hobbies include gardening, reading, and spending time with her family. While she was in the midst of her discoveries, she put a lot of her hobbies on hold in order to completely focus on her research. During the COVID-19 pandemic, she was able to pick them back up again. I admire Jennifer Doudna because of her commitment and passion for her work. She puts all of herself into her research, even putting her hobbies and things that she enjoys on hold. I find that to be really inspiring. She is curious and dedicated to the field of science. I'm inspired by the way she is a female pioneer in a male-dominated field. I also admire the fact that she is aware of the ethical concerns revolving CRISPR technology and advocates for the proper use of it. I think she is a great inspiration for young women, especially those who are pursuing a career in the field of science. Her work in biology has been focused on RNA since her time at Harvard. She is the co-founder of CRISPR-Cas9, which involves genome editing. CRISPR searches for a precise location in targeted DNA, and then an enzyme cuts the DNA in order for a new custom sequence to be added. It is a powerful tool that could cure genetic diseases. Recent research that Jennifer Doudna has been involved in focuses on successfully delivering Cas9 to the right cells in the body without affecting other cells. This study successfully created engineered delivery vehicles that can target specific cells by using antibodies to ensure that the particles would only affect cells expressing the target proteins. They use modified virus-like particles to create vehicles that specifically target the desired cells. Using a single chain variable framing that targets CD19, they were able to show that the Cas9 EDVs had a 74% efficiency with little to no effect on the non-targeted cells. In this diagram, you can see on part A, the antibody that they were targeting, and then part B kind of shows the process of it. And then C through E shows the results of the experiment. In E, you can see the 74.5% efficiency rate. The current research or next steps in the Doudna lab include continuing their discovery and determining the mechanisms of CRISPR-Cas and the associated proteins, such as Cas9, Cas12, and so on. They are also working on developing genome editing tools for use in animals, plants, and in vitro. In addition to that, they are also researching anti-CRISPR genes. Here are my article references.